Hi everyone, this is Aryan. This should also be the seventh lecture in the Introductory Biology series. Today we'll be going over heredity, which is defined as the passing of characteristics from one generation to the next. Basically, how traits are passed from parent to child, and you may intuitively know that if you have tall parents, you'll most likely be tall, or if a family has a history of a certain disease, it's likely that the children in that family will also develop that same disease. However, for traits like height, weight, and even susceptibility to some diseases, genetics aren't the only factor. Generally, lifestyle choices also play a huge role, especially stuff like diet. For example, a good and hearty diet will usually result in reasonable height and weight. On the other hand, malnutrition can actually stunt growth, promote obesity, and increase risk for certain diseases. With that out of the way, let's take a look at a man called Gregor Mendel, who was a monk in the Austrian Empire all the way back in the 19th century. The reason I'm talking about him is because he's often called the father of genetics. Specifically, he taught secondary and university level natural sciences, including math, physics, and biology, until 1856 when he decided to research inheritance, using pea plants as a model organism. Now, a model organism is basically an organism that scientists choose to run experiments on, either because that organism is really easy to work with, or because they model the human body or some other system really well. For example, fruit flies are an extremely common model organism when a lab wants to study genetics, because they reproduce extremely quickly in huge numbers, so you can generate a ton of flies with different mutations across multiple generations within a timescale of less than a month, actually. However, fruit flies really don't model human biology very well because they're, flies. And anatomically, we are extremely different. So scientists that want to study human disease and genetics usually do so using mice or rats. While they aren't as close to human anatomy as chimpanzees, mice are surprisingly close and are roughly 85% genetically identical to humans, I think. Now, the real kicker is that they're way easier to take care of than any other mammal that's this close to humans genetically, and they only have a gestation period or are pregnant for 2 weeks, which is pretty short, adding to the conveniency factor. Anyway, that was a bit of a tangent, but Mendel chose pea plants for his model organism because he had experience with them and they have a few traits that are extremely easy to see. He actually used over 20,000 pea plants in his research, so hopefully that provides some sort of perspective around how much time and energy it took to run all these experiments. Mendel specifically studied discontinuous variation, which is a complicated way of saying that the traits he experimented with didn't affect the expression of one another. He also performed hybridizations with true breeding plants, which is a line with a lot of big words, but basically true breeding plants are plants bred from a set line that always has the same set of traits. And hybridization is really just making two of these true breeding plants reproduce. Also, very specific notation is used to label different generations, specifically P0, F1, and F2. Basically, P0 is the parent generation and are the true bred plants. In the case of this example, the parent generation consists of a pink flowering pea plant and a black flowering pea plant. The next generation, called F1, might be entirely pink flowering, and the next Mendel might have bred together a few of those F1 pink flowering plants to produce the F2 generation, which would consist of 3/4 or 75% pink flowering pea plants and 1/4 or 25% black flowering pea plants. Let's wrap up the summary of Mendel's experiments with a formal definition of traits. Traits are basically just variations in physical appearance of a characteristic. In the case of pea plants, this could be flower color, which can be pink or black, pea color, which can be yellow or green, height, except in the case of pea plants, there are two separate heights instead of a range like for humans, as well as texture, which can be smooth or wrinkly. Now let's move on to two quick probability rules. These aren't too important for the sake of this lecture series, but if you enjoy math and want to do harder genetics problems, they can be very useful. The first one is the product rule, which states that the probability of two independent events occurring is the product of their individual probabilities. So for example, if I knew that pea color and flower color don't influence each other, and I wanted to know what were the odds of having a plant with pink flowers and green peas, I can multiply the odds of having green peas by the odds of having pink flowers. Next, we have the sum rule, which states that for two mutually exclusive events, the probability of at least one of these events occurring is the sum of the two probabilities, or rather of their two individual probabilities. This one is a bit harder to explain until we get farther into the presentation, so just note that I won't really be using these rules, but they can definitely be very useful later on. Okay, now it's time to go over some terms that can sound complicated but are really pretty simple. First of all, phenotype is the observable traits exhibited by an organism. For example, hair color, eye color, and stuff like that are all phenotypes. On the other hand, the genotype is the genetic makeup of an organism. So for example, which genes actually code for hair and eye color. Also, just as a reminder, diploid organisms have 2 copies of every chromosome, and therefore 2 copies of every gene. All the examples we've been dealing with here, or will be dealing with here in this presentation, are diploid organisms, so keep that in mind. Next, we have alleles, which are just slightly different versions of the same gene. So for example, the gene that codes for eye color has an allele for blue eyes and an allele for brown eyes. Same for hair color, maybe they're black, brown, blonde, and so on. Now, homozygous genotype means that a diploid organism has 2 of the same allele. So for example, all humans have 2 copies of the eye color gene in their cells. If you have 2 copies of the blue allele or 2 copies of the brown allele, you would be homozygous for either the blue or brown allele. Now, the opposite of homozygous is heterozygous, which is when you have 2 different alleles as your genotype. So for our eye color example, 1 copy of blue and 1 copy of brown. Finally, the really important thing to remember about alleles is that some are dominant and some are recessive. So whenever the dominant allele is present, it will actually express its phenotype over the recessive, even if the recessive allele is still there. In our eye color example, it turns out brown eyes are the dominant ones, and blue eyes are recessive. So if you had an individual with a heterozygous genotype, meaning 1 blue eye allele and 1 brown eye allele, the dominant brown eyes allele would kind of override the recessive blue eyes allele, and the person would have the brown eyes phenotype, so they would just have brown eyes. All right, the laws of inheritance are 3 rules developed by Mendel after his many years of research, and they're still considered to be mostly true. There are some exceptions that I'll go over later in the presentation. But anyway, let's get started. The first law of inheritance is called the law of dominance, which states that alleles can be dominant or recessive. This is just what I went over. Like some alleles, like brown eyes, will always express themselves over other alleles, like blue eyes. Next, there's the law of segregation, which states that a child is equally likely to inherit either of their parents' alleles. Finally, we have the law of independent assortment, stating that genes do not influence each other's arrangements. This is a rule we'll use later in this lecture when making dihybrid crosses, but you'll see what that is in a few minutes. Okay, let's take a deeper look at the example from the Mendel slide, where the parent generation is a purebred pink flower, or rather purebred pink flowering pea crossed with a purebred black flowering pea, resulting in entirely pink flowering peas in the F1 generation, followed by the F2 generation, which consists of 75% pink and 25% black flowers. Now, before we get started with everything, it's important to define notation. Usually, the dominant allele is uppercase, as you can see at the bottom here, while the recessive allele is lowercase. So, in this case, the gene is flower color, and the dominant allele is uppercase P for pink, while the recessive allele is lowercase b for black. Okay, what I'm going to do is manually calculate the genotype and phenotype if I only had the first generation of purebred peas. Now, we know the genotype for the P0 generation because the purebred, the fact that it's purebred tells us that they are homozygous because they were bred from parents that all had the same trait, so they have to be homozygous. Or not just parents, but a whole line. If it was just parents, we wouldn't know, but the fact that it's an entire line tells us for sure. So anyway, we know that the genotype is uppercase P for the pink flowers and lowercase b for the black flowers. From this point, we want to figure out the genotype of the F1 generation, as that can be used to then figure out the phenotype. And then we can we can determine this genotype by drawing something called a Punnett square. The way Punnett squares work is we put the parents' genomes on two adjacent sides of the square. Or, sorry, not genomes, genotype on two adjacent sides of the square. And from then, we combined the alleles in the actual square to figure out all the possible genotypes. So we'll start by putting the pink genotype, the two uppercase P's, on the left, and the black phenotype, the two lowercase b's, on the top. Now we combine these to get the possibilities of PB, 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 or PB. So all of the F1 generation is going to be heterozygous PB for their genotype. But what does this mean in terms of the phenotype, or actual physical appearance of the flower? Well, remember that if there's a dominant allele, then that will kind of take over the phenotype. And there is a dominant allele in PB, the uppercase P. So the phenotype of the F1 generation will be pink, as you can see. All right, now let's try to figure out the genotype and phenotype of the F2 generation. We will draw another Punnett square, but this time the genotypes are from the F1 generation instead of the P0 generation. And that would mean that both are PB. Now we'll figure out the combinations of allele. In the top left corner is PP, top right is PB, and so is the bottom left, but the bottom right is BB. So now we can clearly define the genotype for the F2 generation, 25% uppercase P, 50% uppercase B, lowercase b, and 25% lowercase b, lowercase b. For phenotype, remember that only homozygous black will be black, because if there's even one dominant allele, the organism will have the dominant phenotype. This means that out of the F2 generation, 75% of our plants will be pink and 25% will be black. As with all subjects, the best way to study biology is to do a ton of practice problems. So we're going to move on to yet another one. This should be a lot quicker. If the eye color gene has two alleles, brown denoted by an uppercase B, that's dominant, to blue denoted by a lowercase b, that's recessive, and the parents have the genotypes lowercase b, and lowercase b, uppercase b, resulting in the phenotype blue and brown colored eyes, what can we expect for the F1 generation? Just like the last slide, we are going to draw a Punnett square and put the parents' genotypes on the side and top. Now we take a look at all the combinations, which are uppercase b, lowercase b, lowercase b, lowercase b, uppercase b, lowercase b, and finally lowercase b, lowercase b. From here, we know that the genotype is 50% uppercase b, lowercase b, and 50% lowercase b, lowercase b. We also know that homozygous recessive double lowercase b will be blue eyes, and the heterozygous will be brown, because remember that the genotype, or rather in the genotype, the dominant allele always trumps the recessive allele. And overall, this results in our phenotype also being half blue, half brown. Okay, so now that we've done some Punnett squares, I'm going to give you the opportunity to practice. And in this case, the gene we'll be dealing with dictates whether a P will be round or wrinkly, with an uppercase R denoting the dominant round allele and a lowercase r denoting the recessive wrinkled one. I'm asking what the genotype will be if two pea plants with the genotypes lowercase r, and uppercase R are crossed. Remember, when doing problems like this, it always helps to draw a Punnett square. All right, I'm going to give you guys a few seconds to work on this, in which you can pause the video and then figure out the answer, and I will go over the correct answer in just a few seconds. All right, so the way we should solve this is by drawing a Punnett square, with one side having the genotype lowercase r, and the other side with the uppercase R genotype. From here, we know that all possible genotypes are uppercase R, lowercase r, and therefore the correct answer is B, 100% uppercase R, lowercase r. Okay, here's a slightly different style of Punnett square. Now we're dealing with the P color gene, which has two alleles, the dominant yellow allele, uppercase Y, and the recessive green allele, lowercase y. We know that two P's were bred, both have a yellow phenotype, and one has a heterozygous genotype. Their progeny has a phenotype of 75% yellow and 25% green. And my question is, what's the genotype of the other P's? Well, first of all, we know that at least one of the alleles in this other P is yellow, because yellow is dominant and the phenotype of this P is yellow. So we could say that the genotype is Y ? Instead of ? or rather uppercase Y question mark. It's important to uh, denote uppercase and lowercase in situations like this. We can, as always, draw a Punnett square, including the question mark as a substitute for, well, the variable that we don't know. Once we combine the alleles, we get uppercase Y question mark, uppercase Y, and lowercase y question mark. Now with this information, and the fact that the phenotype is 75% yellow, 25% green, we can plug in either uppercase Y or lowercase y to see which phenotype matches. Uppercase Y results in a 100% yellow phenotype, while lowercase y gives us the correct fraction. Therefore, the overall answer is that the mystery genotype is uppercase Y, lowercase y. Now we're going to work with a dihybrid cross, which is a way of determining all possible genotypes for not one, but two traits. We can do this according to Mendel's third law, which is at the bottom of the slide here, the law of inheritance, which states, the law of independent assortment, which states that genes do not influence each other's arrangement. We'll be crossing two pea plants with the genotype uppercase R, lowercase y, and lowercase r, uppercase y. Now, in terms of the actual Punnett square, we're going to be using a 4x4 grid and crossing all possible combinations of alleles for both parent organisms. First of all, on the top, the only combinations for the yellow wrinkly pea is lowercase r, uppercase y, while on the left, the possible combinations are uppercase R, lowercase y, and lowercase r, lowercase y. So we'll take every combination to get really only two possible genotypes, just repeated many, many times. And these are uppercase R, uppercase y, lowercase y, and lowercase r, uppercase y, lowercase y. So we know that the phenotype we're looking at are round yellow and wrinkly yellow, because in both cases the dominant yellow allele suppresses the recessive green one, while there's only a dominant round allele in half the progeny. And because our two different possible genotypes are split exactly in half, we know that it's 50% for both of these possible phenotypes as well. All right, now that you have a pretty good understanding of how dihybrid crosses work, here's a slightly harder example we'll be working through. In this case, we're crossing two identical pea plants, both of which are heterozygous for the shape and color allele. So uppercase R, uppercase y, lowercase y for both of them. As always, we line up the possible allele combinations on the sides of the Punnett square, which are going to be uppercase R, uppercase y, uppercase R, lowercase y, lowercase r, uppercase y, and lowercase r, lowercase y for both of them. Now, just to make sure you all understand how dihybrid crosses work, I'm going to go ahead and ask what genotype is going to fit into this space. I'm going to give you a few seconds to work this out. It should not take very long. Then I'll go over the correct answer. All right, so the correct answer to this question is uppercase R, lowercase y, because if we check the parent alleles, or rather combination for that spot, we see that they are both uppercase R, lowercase y. So the only possible resulting combination is uppercase R, lowercase y. Now, the rest of the table, I've also filled that in. I'm not going to go over every single combination, but basically what's important here to remember is the phenotype ratio, which is 9/16 of the peas will be uppercase R, uppercase y, or rather round and yellow. 3/16 will be wrinkly and yellow, and another 3/16 will be round and green, while finally only 1/16 will be green and wrinkly. So when we were going over Mendel's three laws of inheritance, I mentioned that there were some exceptions, and these are called non-Mendelian inheritance. The first type is called incomplete dominance, which is basically when progeny exhibits a blend of both parents' alleles. An example of this is a type of flower called snapdragons. They have a color gene with two alleles, which are red and white, but neither are really dominant. So when you've got a heterozygous snapdragon with the phenotype RW, it actually has pink flowers, which is a mix of red and white. Next, we have codominance, in which both alleles are expressed simultaneously. A great example of this is blood types. You can have one of four blood types, A, B, AB, or O. And if you are A or B, you can only accept the same blood type, or you can accept O. If you're O, you can only accept O blood type, and if you're AB, you can accept any type of blood type, but you can't give any blood. Therefore, O type is called the universal donor and AB the universal acceptor. It turns out that A and B are actually alleles of this blood type gene, and people with AB blood types express both, which is an example of codominance. Multiple alleles are exactly what they sound like. There are cases in which there are more than two alleles. Blood type is actually, once more, a good example of this, as O-type blood is a recessive phenotype, and the O allele for blood type is just a third allele instead of A and B. So A and B are both dominant to the O allele for blood type, which means you need to have two O alleles to have it. Oops. All right, I'm going to quickly provide some details about chromosomes, as I really touched on them really briefly in previous lectures, and we need a more complete understanding to learn about genetics in detail. So we have 46 chromosomes, of which 44 are autosomes, or non-sex chromosomes. This means that every single human cell has these same 44 chromosomes or rather, every single human has the same 44 chromosomes with some degree of variation. The other two chromosomes are called sex chromosomes and are either X or Y. They're called sex chromosomes because they determine biological sex, where XX is female and XY is male. All of the traits I've talked about so far are found on autosomes, which means that all people have two genes, therefore two alleles for them. But traits can also be on the X chromosome. In that case, any male with the gene would have to express it, even if it's recessive, because they don't have another X chromosome. Remember, their sex genotype is XY. And yeah, so we're going to take a deeper look at X-linked traits in the next slide. Also, mutations are changes in the genome. They can be negative, meaning that they hurt the organism, silent, meaning that they make no difference, or very rarely positive, meaning that they actually benefit the organism. Additionally, on the bottom right of the slide, I have a picture of the 23 chromosomes here from 1 to 22, and then X and Y. That's just for reference of how they vary in size. Okay, now we're going to take a look at what a Punnett square is for an X-linked trait. Here we have two alleles, XN and XM, where XN means normal, so X normal, while XM means a mutation, so X mutated. And the mutation is recessive, while the normal allele is dominant. Now, I want to know what the phenotype and genotype is for these children of these two parents, one with a genotype of XMY and another with the genotype of XNXM. So basically that's a male with a mutated allele and also a mutated phenotype crossed with a female that has a copy of that mutated allele, but not the phenotype because the normal allele is dominant. The notation is a bit more convoluted, but other than that, this will be exactly the same as the other crosses we've done. So let's draw this Punnett square. As always, the sides will be genotypes, specifically XMY and XNXM. So the resulting combinations will be XNXM, XMXM, XNY, and XMY. So basically, a 25% chance for one of four unique genotypes. However, the phenotype is split 50-50 because the XM, XM, and XMY will have the mutation. Will have the mutated phenotype, while XNXM and XNY will not. All right, now we're going to apply what you just learned to a practice problem. Color blindness is an X-linked disorder, which means that the odds that a male child will be colorblind if his father is and his mother is carrying the gene, but is not colorblind. Remember that I'm asking about male children, and the best way to do these problems is with a Punnett square. So I'll give you a few seconds to work through this, then you can pause the video, and then I'll go over the answer. Okay, so this is the relevant Punnett square. It's identical to the one that we just did, as you can see for males. The genotype with a Y chromosome, there is really only one of them with the mutated X allele and one with the normal X allele. So we can say that they have a 50% mutated X phenotype. Therefore, the correct answer is C. Once more, it is really important to pay attention to the question. So I was asking not about the overall phenotype, but about the male phenotype. Okay, now I'm going to be going over a series of human genetic disorders. So all of these are diseases resulting from mutations in a gene or a few genes. First of all, phenylketonuria, or PKU, is an autosomal recessive disorder, which remember means that its gene is found in a non-sex chromosome plus it's recessive. It happens when the amino acid phenylalanine cannot be broken down. Compared to the other disorders we're going to go over, it's relatively easy to treat. Basically, the person or anybody who has this will have to avoid foods containing phenylalanine and just not have anything in their diet, or else there is a high risk for serious mental retardation. Next is cystic fibrosis, which is the most common lethal genetic disease in the United States. Essentially, in it, there is a buildup of mucus in organs like the digestive tract and lungs. And turns out roughly 1 in 25 Caucasians are a carrier for it. Next, we have sickle cell anemia, in which there's a mutation in the gene coding for proteins, or rather for the hemoglobin protein, which normally carries oxygen in the blood. This mutation results in red blood cells shaped like crescents or sickles, but also, interestingly, it confers resistance to the disease malaria. There's actually a theory that because sickle cell anemia is found primarily in people of African descent, that it may have evolved because individuals with the mutation were far less likely to die of malaria, even if it did cause long-term issues. Now, Tay-Sachs disease results from a mutation in an enzyme that normally breaks down lipids, which can cause their buildup in the brain, which may cause actually a lot of issues, including seizures, blindness, and potentially death. Huntington's is different because it's autosomal dominant, so just one mutated copy is sufficient for the disorder phenotype. Huntington's is a neurodegenerative disease in which certain pathways degrade beginning in middle age, almost always resulting in death eventually. The first X-linked disease I'll be going over is color blindness, in which certain colors are harder or impossible to discern. I actually have a mild version of red-green color blindness. It's by far the most common and least dangerous disorder on this list. Finally, hemophilia is a disorder in which blood cannot clot correctly. And these are just meant to be some examples of disorders that are directly a result of mutations. There is by no means a need to memorize this list. So all those diseases I just showed you were caused by just a few mutations. But there can also be mutations that affect large swaths of a chromosome or even more than one chromosome. The first one is called a deletion, which is when a part of the chromosome is, deleted. There are also inversions, which is when a part of the chromosome is reversed. So if you can imagine the series of genes which were in that region would be read backwards, which is pretty much always lethal. Now, a translocation is where a part of one chromosome is basically removed by accident and attached randomly to a different part of a different chromosome. In polyploidy, there is at least one complete extra set of chromosomes. For example, humans have 2 times 23 or 46 chromosomes. So if they were to have 3 complete copies of all the chromosomes, it would be 69 or 3 times 23. And if it were 4, it would be 4 times 23 or 92, etc. And it turns out that much of the fruits we eat are actually polyploid because it makes them significantly larger, but I'm not sure if it's possible for animals to survive being polyploid. Okay, our next mutation is a non-disjunction, which is when 2 homologous chromosomes don't separate completely, which can result in embryos with one or more chromosomes, with one less or one more chromosomes than normal. And these are called trisomies and aneuploidy, if there are 3 of one chromosome or only one of a chromosome, respectively. All right, so there are only 3 big human chromosomal disorders because in almost every other case of a chromosomal mutation, the child will die. The child will die either really early in their life or before even being born. So anyway, the first one is Klinefelter syndrome, in which the X chromosome is duplicated. So there is an X and a Y. So XXY is the genotype. The presence of this Y chromosome means that the people with Klinefelters are male, but they do have some reproductive issues. There's also Turner syndrome, which is denoted by XO, because in it there is only one X chromosome. And people with Turner syndrome are generally considered to be female because they have no Y chromosome, but once more, there are quite a few reproductive issues. Finally, Down syndrome is trisomy 21, which, remember, means that there are 3 copies of chromosome 21 and comes with a series of symptoms and heightened risks for other diseases. With that in mind, Down syndrome is a result of which of the following? I'll give you a few seconds to answer, and remember back to 2 slides ago when I was talking about all the different types of chromosomal mutations. Alright, so the correct answer here is a non-disjunction, because Down syndrome is an example of a trisomy. It's trisomy 21. And trisomies are always the result of a non-disjunction. Okay, now we're going to go over a brief summary of the important terms covered in this lecture. First of all, a trait is the version in, the variation in physical, a trait is the variation in physical appearance of a characteristic. Alleles are slightly different versions of a gene. A phenotype is an observable trait exhibited by an organism while the genotype is the organism's genetic makeup. A homozygous genotype means 2 copies of the same allele, while a heterozygous genotype is 2 different alleles. Incomplete dominance is when progeny exhibit a blend of parents' traits, like snapdragons. Codominance is when 2 alleles are expressed simultaneously, like the AB blood type, and multiple alleles are when there are more than 2 alleles, like the ABO blood type system. All right, let's finish up with this. Mendel developed the 3 laws of inheritance, which were dominance, segregation, and independent assortment. Also, I've included the types of chromosomal mutations here, but because I went over them pretty recently, I'm not going to go over them again. Anyway, that just about wraps it up for the introductory biology lecture number 7 on heredity. Next time we'll be going over evolution.